Hello, everybody, and welcome back to Creation Myths. Today, we are talking about how Discovery Institute has accidentally refuted intelligent design. There's a bit of a backstory here that we have to go through in order to get to our final destination today, but this all starts with my debate with Dr. Casey Luskin from way back on May 2nd of 2024. This debate was about the human genome and junk DNA. The topic was, is the human genome largely junk DNA? Since that debate, I've been having a conversation with Discovery Institute. They've published on their Evolution News blog at least 9 articles about that debate, responding to the debate, related to the debate, mentioning me in the context of that debate or our ongoing back and forth following the debate. So here are all those articles. If you want to look at those titles, you can look those up if you want to read kind of their output related to this conversation following that debate. But over the course of this conversation, a really interesting thing has happened because this conversation isn't just about junk DNA anymore. We've gotten out of that specific realm into kind of topics that have much broader implications. So that's what we are going to talk about today in trying to defend their position on junk DNA, the position that the human genome has very little, if any, non-functional DNA. Discovery Institute has made arguments with broader implications for intelligent design as a whole. So today, we're going to talk about those arguments and those implications. We really got here through this question that I asked during the debate and have been asking since. The question is this: 92% of the human genome does not experience purifying selection. What sequence-independent functions are these unconstrained regions doing? For weeks, I've gotten no specific answer to this question, but now I've gotten an answer from Discovery Institute. Sort of. This came up via a Twitter conversation of all things with David Klinghoffer, who is a Discovery Institute fellow at the Center for Science and Culture. It started with this piece that he wrote called DNA Repair and the Origin of Life, Here's the Problem, in which case he took what I think was a bit of a gratuitous swipe at me. But you know what? Hey, all's fair. It's cool. So what I did, because, you give it right back, is I went on Twitter and said, hey, maybe David Klinghoffer can answer, since Drs. Luskin and McClatchy, that's Dr. McClatchy, the other Discovery Institute, he's actually an evolutionary biologist that I've been going back and forth with. Maybe David Klinghoffer can answer, since Drs. Luskin and McClatchy haven't, what sequence-independent functions are found in the 92% of the human genome that doesn't experience purifying selection. What resulted was kind of a long Twitter back and forth with David Klinghoffer. He answered, the myth that 92% of the genome must be non-functional because it isn't conserved was answered long ago by intelligent design proponents. Here are a few links. Now, I pointed out those papers are not relevant and clarified what I'm actually doing. So my response ended with this. After it was a little 4-tweet thread, I said the claim from ID proponents is that the vast majority of the human genome is functional. Most of the genome is unconstrained at the sequence level. Therefore, most of those purported functions must be sequence independent. I'm asking, what are those specific functions? Right? So I'm clarifying what I'm doing here. I'm not making an affirmative claim that these regions are non-functional. I'm saying if they are functional, those functions must be sequence independent. What are those functions? David Klinghoffer responded, unconstrained sequences don't mean the function is not sequence dependent. Dr. Dan doesn't know how to not think like an evolutionist. And there are many sequence-independent functions known for DNA, such as locations of centromeres and kinetochores. Now again, I responded to this. I asked a question. I said, is it your contention that specific sequences can be maintained in the absence of purifying selection? And a little bit of time went by and he didn't answer, so I pinged him again. I said, if it is true, it was a short thread, it was just 3 bits. I'm reading you the second 2 because the first one just quoted that question. I said, if it is true, as David claims, that unconstrained sequences can have sequence-dependent functions, then it must be the case that A, they maintain those sequences absent purifying selection, or B, many sequences can do each specific biochemical function. I went on to say, option A, flies in the face of population genetics. Option B undercuts a central premise of intelligent design creationism. So which is it? Discovery Institute, that's the Twitter handle for Discovery Center for Science and Culture. Okay, so that was my exchange on Twitter with David Klinghoffer, which led to this article on Evolution News titled Dr. Dan Has a Taste for Debate. In this piece, David Klinghoffer wrote, quote, "From what I've been told, there are many examples of non-conserved regions having sequence-independent functions." To clarify, those are functions that are not dependent on a specific DNA sequence. Compare 2 strains of yeast, and they will often have almost no sequence identity between centromeric regions. Yet those regions are essential because this is where kinetochore is formed and spindle attachment occurs. There are many other examples. Is it not so? He's going to answer that question subsequently, right? But at the end of this particular article, I thought this was really funny. He said, by the way, we've put out like other videos on other topics since then. A new one is out now. Perhaps it's time to move on. And just a brief aside, I will just say this to Discovery Institute. No, I'm not going to move on from this topic. 1, I'm having fun. And 2, we're learning a lot here together, I think. So no, I'm not going to move on to this topic. We're going to continue having this conversation as long as you're going to keep responding to me, which I hope you keep doing. Moving on, then after this, they published a second article. The byline for this one was just Evolution News. So it wasn't David Klinghoffer, it wasn't Casey Luskin, it wasn't McClatchy, it wasn't anybody. It was just Evolution News. And they provided documentation for Dr. Dan. And they start by quoting this paragraph that we saw in the previous article that ended with, "Is it not so?" and said, "It is so. Here's some documentation." Right? And they provided a bunch of references having to do with how centromeres exhibit lots of different sequences, right? So they seem like the sequence, if you compare different species, doesn't actually matter all that much to the function of being a centromere. And they ended by saying, "We hope Professor Stern Cardinal finds these citations of interest. And let me tell you, Discovery Institute, I certainly do. I appreciate this more than you realize." Now, first, before we get to the broader implications of what Discovery Institute is doing here, I want to point out they're still wrong when it comes to junk DNA. These functions that they say are sequence independent, like being a centromere, these are not actually sequence-independent functions. They're constrained within each species, even though each species uses different sequences to do this particular job. And for evidence of this, we can go to this paper from Journal of Biology, Who's Driving the Centromere?, in which we learn that despite the fact that centromere satellites are so divergent between species, they show a high level of sequence conservation between the repeats within a species. So we have lots of divergence between the species, but within each species we see consistency. In other words, we see constraint. They're subject to purifying selection within each species, even though there's a lot of variation between species where different sequences can all do this same job. So on the narrow point here, right, is the evidence they're providing of centromeres having all these different sequences being evidence of functions that are sequence independent? No, that's not what they're doing. They're not actually providing evidence for the thing they think they're providing evidence for. Also, all of the things they're providing, they're all the same thing. They're all centromeres. And we all agree that centromeres are functional. Way back in my debate with Dr. Luskin, I went through like bing, 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 here are the functional parts of the genome and the percentages. Centromeres were already in the functional column. So this doesn't even come close to answering the question of what is that 92% of the genome doing? What are the specific functions we're going to find? If they had better evidence than this in those like 800 papers showing that junk DNA is actually functional that they provided before our debate, if they had better evidence, if they had a better answer to that question in those papers, we'd have seen it by now. I don't think they have the goods on this one, folks. I don't think they have an answer at all. Maybe they'll surprise me. In fact, I hope they surprise me. Discovery Institute, I'm going to keep asking. I hope you can give me a better answer to this question that I asked during the debate and have been asking since. And this is the best answer you've given, and it's not a good answer. I hope you keep trying. Give me a better answer to this question, please. But again, this is kind of a smaller point that I want to make with this particular video. Because you know what? That's okay. I understand the broader point they're trying to make, which I might summarize like this. There are a number of sequences that can do this or that biochemical function, many of them currently unknown. So a lack of purifying selection and or signatures of known functional sequences does not preclude a particular region from doing a particular function. I think that is a fair representation of kind of the larger scale argument that Discovery Institute is trying to make here when it comes to identifying regions of the genome that are functional, that might be functional, or that are almost certainly not functional. I think this is an accurate representation of the point they are making. Discovery Institute, feel free to correct me if you feel like this is an unfair representation of your argument, but believe me, I am trying to accurately represent the argument you are making. Dr. Casey Luskin, agrees with what I just wrote as far as I can tell. He actually alluded to this during the debate. Take a listen to this moment right here, where here he makes what I think is basically that same point. Take a listen.
But I do know that there's lots of functions for non-coding DNA that do not require something to perform its normal quote unquote function, providing a transcript that can actually be processed or whatever. You can have, attachment points for promoters, enhancers, et cetera, et cetera. And I just don't see why. And these can take all kinds of different sequences.
Now, before I respond to this point, I want to make a couple of assumptions crystal clear so we're all on the same page and everybody knows where I'm coming from. My first assumption here is that everybody at Discovery Institute involved in this conversation understands the question that I'm asking, right? The question being, what functions do we find in that 92% of the genome that's not sequence constrained? Therefore, what functions can we find that are sequence independent? I think everybody understands kind of the parameters of that question. And then my second assumption is that the folks at Discovery Institute are making a good faith argument in response to answer that question that they believe is factually accurate and logically valid. I don't think they're making a bad faith argument here. I don't think they're just grasping at straws trying to come up with the first thing that they think makes sense. I think they have gone into the research. They have pulled out what they think is both an accurate representation of what we know and also answers my challenge. Okay, so I think they're making a good faith argument that they think is accurate. So I want to be clear about these two assumptions. I think I am on firm ground with both of them. So now with that being said, let me get to my response. With regard to their broader point having to do with functional sequences, my response is this. Their broader point is correct. They're basically right that a lot of different sequences can do different functions. And there's a lot of data to this effect, right? Here are two things we know based on direct observation in, from experiments and collecting data and looking for functional sequences. First, that there are often many sequences that do a given function approximately equally well. And second, it's likely that many potentially functional sequences are unknown. We know this because when we do experiments, we find sequences that are able to do functions that do not exist in nature. We find new sequences to do the same functions. So again, with regard to their broader point in terms of the frequency of functional sequences and the ability of different sequences that are quite different from each other to do specific functions, I think Discovery Institute is basically correct here. And here are some examples of this from the literature. So from 2003, we have this paper, Experimental Prediction of the Evolution of Cefepime Resistance from the CMY2 AMP-C beta-lactamase. This study was really interesting. They actually discovered two completely novel antibiotic resistance alleles that had never been found before in nature. And we have NEMI 2017, random sequences are an abundant source of bioactive RNAs or peptides. So basically you just generate a bunch of random either long non-coding RNAs or actual protein coding sequences and you get new peptides. And in this paper they found one of each, long non-coding RNA and a polypeptide that were adaptive, just completely brand new from random sequences, in addition to a whole bunch of other molecules that had less robust evidence for their benefit, but still evidence of some biochemical activity. Then we've got Yona et al. 2018, random sequences rapidly evolve into de novo promoters. In this study, they did a really interesting thing. They looked at just random sequences of 100 nucleotides and asked, if we generate random sequences, can they function as promoters in bacterial cells? Turns out the answer is yes. And many of these random sequences had activity as a promoter that was equal to or even exceeded the promoter activity of the wild type promoter. And then finally, more recent one, 2023, Frumkin et al. Selection of a de novo gene that can promote survival of Escherichia coli by modulating protein homeostasis pathways. Basically, again, we're just trying out random sequences, and what we find is you could actually have a selection for a completely new gene that was randomly generated. Okay, so the takeaways from these are just 4 examples of what is a robust body of work. The takeaways are that you can have biochemical functions where we know that this sequence or that sequence does the job in nature. We can find lots of other sequences that will do those jobs when we look for them that aren't present in nature. So why does this matter? Why do we care about all of this stuff? To answer that question, we have to look at intelligent design more broadly. So I want to direct your attention to this piece from 2015, An Introduction to Intelligent Design by Casey Luskin. In this piece, Casey Luskin wrote, "No physical or chemical law dictates the order of the nucleotide bases in our DNA, and the sequences are highly improbable and complex. Yet the coding regions of DNA exhibit very unlikely sequential arrangements of bases that match the precise pattern necessary to produce functional proteins. Experiments have found that the sequence of nucleotide bases in our DNA must be extremely precise in order to generate a functional protein. The odds of a random sequence of amino acids generating a functional protein is less than 1 in 10 to the 70th power. In other words, our DNA contains high CSI, which is short for complex specified information. And this is like the design test, is does a particular thing have complex specified information? Is it complex? Is it unlikely, right? Is it an unexpected pattern? And is it highly specified? And if you read this article, Casey Luskin talks about 2 mountains, right? He says, if you look at this mountain, which is just a plain old mountain, yes, its specific conformation of rocks is highly unlikely. It's unlikely to have the exact shape that it has. No other mountain is shaped exactly like this, but it is not specified. It's just a plain old mountain. Therefore, we don't have to attribute its specific shape to design. Compare that to Mount Rushmore, which is equally unlikely statistically in terms of its conformation, but its conformation is also highly specified, right? It has 4 faces built into it. So that allows us to attribute design to Mount Rushmore, but not, this other plain Jane mountain that doesn't have a giant sculpture on it. The idea here is that we could apply these same criteria to our genome. Any given sequence of nucleotides is going to be highly improbable, right, and therefore complex, right, whether it's 100 nucleotides or 1,000 or 10,000. Yeah, that specific sequence is going to be rare, but is it specified? Does it have the particular sequence to do a particular job, right? That's the specified part of the equation, and without both of those parts, you fail the design test, right? You don't meet the criteria for attributing design. In other words, we can conclude, applying this to the genome, we can conclude that the genome is designed because functional sequences are both highly unlikely and highly specific. Now, the paragraph I just read to you was specifically for protein coding sequences, but if you look at Discovery Institute's output, they apply the same logic and these same criteria to non-protein coding functions. So they're talking frequently, for example, about regulatory functions. The same criteria apply. But here's the thing. David Klinghoffer and Dr. Luskin are now arguing that you don't need highly specific sequences to be functional. They're actually correctly arguing that a lot of different sequences are capable of doing particular functions, even sequences that we are currently unaware of, which means these functional regions that neither exhibit nor require one specific nucleotide sequence or one of a very few to do their job would fail to meet their own design criteria. The criteria are complex and specified. And once you open the door to sequence independent functions, you've removed the specified part from that equation. This undercuts the central premise of intelligent design. If many functions can be accomplished in a sequence-independent way, which is what David Klinghoffer has argued, he said right there, is it not so, that you can find lots of functions that don't require specific sequences, lots of functions that are sequence independent, then we've removed the specified part from complex specified information. We've removed from our functional nucleotide sequences one of the two pillars of design detection according to Discovery Institute, and specifically according to Casey Luskin. And if there are many potentially functional sequences, which there are, which Discovery Institute has acknowledged is the case, then they are neither as rare nor as unlikely as intelligent design proponents would have you believe. For example, Douglas Axe's 2004 paper, which means these functional sequences neither meet Discovery Institute's criteria for design nor require design as an explanation. And I want to be crystal clear about this. This is due to arguments being made to me by intelligent design proponents. These are the consequences of the arguments they are making to me in service of the argument that the human genome is largely functional and has little, if any, junk DNA. So in conclusion, in arguing that most of the human genome is functional, intelligent design proponents have correctly taken the position that potentially functional sequences can be diverse and are often not required to adhere to a single specific sequence or a small number of specific sequences. This undercuts the central argument of intelligent design creationism, which is premised on the idea that functional sequences are both extremely rare and highly specific. But now, they acknowledge, they're in fact arguing, that functional sequences can be diverse and are often unknown, which means they can be more common than we thought. Even though they miss their mark, specifically with junk DNA for reasons I explained earlier, these points are consistent with available data, and I compliment Discovery Institute for that. And I also compliment Discovery Institute for making a very strong case against intelligent design. Thank you for watching. Please remember to hit like before you leave, subscribe to the channel if you're here for the first time and aren't a subscriber, leave a comment, and share this video. And if you want early access to my pre-recorded videos, please consider becoming a channel member. Thank you in particular to my super subscribers, my second tier members. Thank you all again for watching, and as always, don't get fooled.